I am Anton Lord, and I am a postdoctoral research fellow at the Leibniz Institute for Neurobiology. We use magnetic resonance imaging to measure metabolites that affect brain function, such as glutamate. Anomalies in glutamate concentrations and communication between brain areas are common in patients with depression. We wish to understand how these measures are related to depression so that they can be used as markers in the clinic.
Hello, my name is Leonardo. I am a PhD student in the project. What we do here is we are using magnetic resonance imaging, an non-invasive imaging tool, to assess the impact of depression on the brain structure and function.
Structural MRI scans allow us to assess the shape and the integrity of brain areas. Diffusion scans allow us to observe the white matter fiber tracts which connect each region with other regions. Magnetic resonance spectroscopy allows us to measure the concentration of metabolites and neurotransmitters in brain areas. Functional MRI detects brain activity and how the different brain areas interact with each other.
I'm Fionn Dunphy-Doherty. I'm a PhD student at the University of Nottingham. Our main focus is to investigate how depression affects neurons in the hippocampus, a brain region that's involved in learning and memory and also emotional regulation. The hippocampus is especially vulnerable to depression, and the disease can lead to reduced neuronal growth and integrity in this region. Our goal is to develop MRI markers that can be used in humans to evaluate the extent and the progression of this damage and its potential regression following successful treatment. Here we can see how neurogenesis is measured using different stains of the dentate gyrus. The green stain shows nu-N, which is a marker for neurons, while the red stain shows B/RDU, which is a marker for proliferating cells.
So hi, I'm Jennifer. I'm from France, from Bordeaux, and I'm studying here my PhD in Trinity College Dublin. I'm specifically investigating how astrocytic-like cells are affected in depression. Astrocytes are cells that support neurons in the brain. These cells regulate blood flow and are able to remove the glutamate that normally accumulates in synapses. Glutamate is an excitatory neurotransmitter that in excessive quantities can be toxic to neurons. And our hypothesis is that depression is associated with associative dysfunction in areas of the brain that are of key importance in emotional regulation, leading to an increase of glutamate and ultimately to the integrity of neurons in those regions. By making astrocytes illuminate under fluorescent light, it is possible to observe them under the microscope and assess them in their structure, number, and function.
Hello, my name is Alison McIntosh. I'm originally from Scotland and I'm a postdoctoral researcher working here at Trinity College Dublin. The project that I'm working on aims to establish neuroimaging markers related to depression and antidepressant action in our animal model. We are using a multimodal magnetic resonance imaging approach to study changes in brain structure and function, along with cerebral blood flow and alterations in specific metabolite concentrations. One key benefit of using animal models in depression research is the ability to combine behaviour with looking at molecular markers in brain tissue, coupled with performing the same MRI scans in animals as you would perform in the clinical population.